Thank you, Margie, for that introduction. Can everybody hear me? I'm going to try and stay close to the microphone. I do tend to wander when I talk, so I'm going to try really hard and stay by the mic. If I go too far away, just wave your hand, and I'll know that you can't hear me, and I'll come back. So I'm, as Margie said, I'm Melanie McConnell. I am a cancer cell biologist, and my focus for the last 10 years really has been on brain cancer, so the neuro-oncology. And the work I want to talk to you about was indeed funded by the Neurological Foundation, and it's built on a lot of work we've done over the years. So before I do anything else, I need to acknowledge the people that did the work. I sit in my office and I have crazy ideas, and these people are the ones that have to try and translate them. So this is my team at the moment. We're a smallish group. There's about 7 of us at the moment. And Devlin Forsythe, who you're going to hear from later, it's his work really. It's all of his work that's driven this project that I'm going to talk about. He took a really crazy idea that I had and he actually made it work. And it's taken 5 years, but he's done it, and I'm really, really proud of it. Okay, so this is our other introduction. This is glioblastoma. Does anyone in the room know anyone with a glioblastoma who has had a glioblastoma? Yeah, there's a few of you out there. It's a really horrible, horrible disease. Do I have a laser? I have a forward and a backward. All right, I have no laser. I'm going to stand still and wave my arms. So this is a really aggressive primary brain cancer. It's what happens when a mutation or a mistake happens inside a brain cell, and that brain cell starts to proliferate and to grow and expand when it shouldn't. And they're not uncommon. You know, there's 4 or 5 people in the room who know someone who've had them. I know people who've had them, and they are kind of nasty. This is a picture of a patient we had here in Wellington, and we have the hole. Can you see the hole without me moving? So that was where the primary tumour was removed, and then this person had 6 weeks of radiation therapy and 6 months of chemotherapy. And while they were having their treatment, the tumour cells just kept on growing and growing and growing. So we can see from looking at this that the therapy did not work. Those cells continued to grow despite the fact we were throwing everything that we have at them. And so what we end up with, when someone's diagnosed with one of these, the average length of time that they'll live past their diagnosis is 15 to 18 months. And there's about 5% of people who'll live out to 5 years. There's always a small number of people who live much longer, and we don't understand what's different about the disease in those people, but there's always what we call this long tail. So I'm going to show you a few of these graphs. This is showing you people at the time of diagnosis, and then how long they, in this case, how long they lived for. And so we have some people who, there's always these people out here. So there's something about this disease that means that it's not a simple disease. Some people don't live very long, and some people might live 5 or 10 years. I think 16 years is the current record that I'm aware of in New Zealand. So it's a curious disease. So I'm now going to give you an abbreviated version of my second year cells lecture that I give up at Victoria University. So I hope you've all got your notebooks with you, because there'll be a quiz at the end. Okay. Someone loves taking, you've actually got paper with you. This is why we've given you a pad. Okay, so a brain cancer is a malignant cell, and there's a lovely quote, and this comes from the textbook that I use in my second year class, and it says, cancer cells break the most basic rules of cell behaviour, and they exploit every opportunity to do so. And I kind of like this because it gives us this adversary. It's kind of giving personality to these tumours. And there's lots of different kinds of brain cancers. There's 130 different types of brain cancer. And some of these are benign, and some of them are malignant. Some of them are of brain origin, and some have come from somewhere else in the body, and they're growing in the brain. But the focus of the work today is going to be on the malignant tumours that started in the brain. So here's a very basic kind of schematic of what a tumour looks like. And so when we think about what's in a tumour or a cancer, I'm going to use those interchangeably. There's a slight difference in what they mean, but I'm not going to quiz you on that. So I will use them interchangeably. But essentially there's three major components to any kind of tumour. The first thing we have is the tumour cells themselves. And these are the ones that have got the mutations in them. These are the ones that have got the growth advantage. They grow and they expand. Around those, we have what we call stromal cells. And so this is things like the blood vessels that feed the tumour. And there's other cells in there, normal cells, that actually support the growth of that tumour. And then the third component is the immune cells. And so some of those are tumour-fighting immune cells, and some of those are actually tumour-supporting immune cells. There's a fourth component that everyone forgets about, which is the matrix is the glue that holds everything together. So a cancer is a really complex mixture of all these different parts. So how do we fight a brain cancer? Well, we've got three major targets. We can attack the tumour cells. So these are the ones that have those mutations, and you'd be familiar with things like radiation therapy. It's a mainstay of brain cancer treatment. And then we also have some chemotherapies that we can give, drugs like temozolomide or Temodar. If you have a low-grade tumour, you might be very lucky and be able to get hold of this drug here, vorasidenib. So these are the therapies that attack the tumour cells themselves. And they are variously successful or not, depending on the nature of your tumour. We have therapies that target those stromal cells. So we can target the tumour cell, or we can target the stromal cells around the tumour. And some people would be aware of this treatment Avastin. Very frustratingly, Pharmac decided not to fund this for brain cancer patients, despite the fact that it improves the quality of life of patients enormously. We're still fighting that decision. We are still debating that decision, pressuring Pharmac to try and fund this drug, because it targets those stromal cells. It's not, you can do it in combination, and then you can hit the tumour and the stroma, and then you've got those sort of 2 out of 3 angles. And then the last one is we can try and fight using the immune cells. And this is a picture of a cancer cell in blue. This is I believe it's a computer-generated picture of a cancer cell. I don't think they're actually blue and shiny. But it's a representation of a blood cancer cell, and it's being targeted by all of these immune cells, which are the purple ones. So that brings us to the topic tonight, which is immune therapy. Now, there's 2 different ways we think about this phrase immune therapy. Some people take it to mean a therapy where we're using a product of the immune system. And so there's a lot of treatments for diseases that are made of monoclonal antibodies. But what we're really interested in is therapies that actually modulate the activity of the immune cell. And so these might be therapies that enhance those natural tumour-fighting cells, and we call those immune checkpoint inhibitors. You'll be tested on that phrase as well. And then we can also introduce engineered T cells, tumour-fighting cells, and these are the CAR T cells that some of you might have heard of. Okay, so the immune therapies that we're talking about, they all harness the power of T cells. So the T cells are a really powerful cell in your immune system. And what a lot of cancer immune therapies try to do is to enhance the natural ability of the T cell to fight the tumours. And so there's these 2 drugs. One of them's called 3 drugs. There's Yervoy, which isn't very popular these days, but there's a drug called Keytruda, and there's a drug called Opdivo. A lot of you have heard of Keytruda? Yeah, so Keytruda is one of these compounds, and what it does is it tries to enhance the activity of those tumour cells. It's being funded now for a whole bunch of different cancers in New Zealand, which is amazing. And so what this tries to do, so T cells, first of all, you have to turn the T cell on. And so the T cells are activated by other cells, and we call them surveillance cells or antigen-presenting cells is their correct name. And so the antigen-presenting cell says, hey, I saw a tumour in the environment. It looks a bit like this, and it educates the T cell as to what's going on there. And then that activated T cell will recognise that tumour cell and it will kill it. And so we have the on switches for the T cells, and then we have the off switches. And so what the cancer cell likes to do is to avoid being targeted. So the only reason that we get cancer is that our T cells failed to stop it. So cancers form because they can stop the T cells. And so they can hide so that they can't be spotted. They can stop the T cells from being activated by ramping up the kind of off, by turning up the off switches on those T cells, and they can also stop the T cell from recognizing them and being able to act. And our immune checkpoint inhibitors, they act on these switches. So sometimes if the tumor cell is trying to turn off the T cell, we can turn it back on again using these drugs. And so that's what Keytruda does. It helps to turn the T cell back on again. And so this can be really effective. Now, this is an example of melanoma, and the first trials for these drugs were in advanced melanoma. So metastatic melanoma, it spread through the body. Until 2011 or 2012, your chance of living beyond 12 months with a metastatic melanoma was pretty low. It was an untreatable, uncurable disease. When we start using these checkpoint inhibitors, the ones that allow the T cells to come back on, we can see that we get some really dramatic results. So this is real world data. This was from a hospital in Israel, and they said, we went back and we looked at our records, and what they found was 56% of people who had advanced melanoma had their disease held stable or even cured by these checkpoints, by drugs like Keytruda. You can see that 44% of people didn't, so we don't really know how to use these properly yet, but you can see that you've got a pretty good chance of having your disease either completely cured or stopped in its tracks by these drugs. And it's not just melanoma now. We know that lung cancer, kidney cancer, bladder cancer, liver cancer, bowel cancer, throat, breast, some of the lymphomas, mesothelioma, these all respond really well to having the T cells turned back on again. And it's only ever a subset of people, so there's always like a 50-50 chance that it will work for you or it won't, but 50-50 is pretty good, really. 60-40 maybe, 70-30 if you're really lucky in those cancers. So obviously, this has been the question for us. Can we do this in these brain tumours? Because we don't have anything that works in our brain tumours. And so the very first studies were done in melanoma. The very next study that was done with these drugs was in brain cancer. And that was in 2011, that study, that clinical trial was started. Have you heard about the success of this treatment? No. We haven't heard about the success of this treatment. So I've picked out a couple of examples. There's been trial after trial after trial, and they all have the same results. So this trial, they put 80 people in to get the checkpoint inhibitor. They compared it to just chemotherapy alone. No evidence of any difference between the life expectancy. The addition of ipilimumab, which is the long name they give the drug, did not show an effect. No further trials are planned. And so that has come out of trial after trial after trial. Here's another example. You can see that this is our survival of our patients. Everyone's alive at the beginning of the trial, and slowly they pass away. And the grey line is temozolomide, which is our most effective treatment. 10% of people are still alive at 33 months. The immune therapy actually killed people faster. So it's very disappointing. It's been a decade now of trying to figure out, well, maybe we had the wrong combination, maybe we need to do it differently, maybe something, people are still doing these trials. This is our latest form of immune therapy. This is our engineered CAR T cells. So the Malaghan Institute here in Wellington are running an engineered CAR T cell trial. It's basically you genetically engineer the person's T cells and you put them back in and they're super revved up and they'll hit the tumour. Really successful in the blood cancers. Really successful. And so this is David Downs. You might have seen him on television. He did a series in Stuff where he called it a mild touch of the cancer. He wrote a column in Stuff where he thought he was going to die of his untreatable lymphoma. He went to the US, he mortgaged his house, he spent more than $1 million. He got these engineered T cells, and he's now alive and thriving, and he's one of the on the board at the Malaghan Institute now. So these work really well, again, in about half of all patients. And there have been some trials of this in brain cancers, but again, nothing effective, nothing long-term, nothing durable. We're seeing a little bit of a hint that there's some immune cell activation, but they can't do anything. So this was my question. What are we missing? We are missing something. Now, there could be a prize for the person who guesses what it is that we are missing. Do you remember the earlier slide? Tumours are full of immune cells whose job it is to help the tumour out. And we've kind of forgotten about these tumour-enhancing cells. They're really important. And so particularly those brain cancers, they don't have an immune reaction sitting there waiting for the T cells to be released. There's nothing ready to spring into action. And it's because they're full of these immune cells that can really block, they block any immune reaction. And so all of the preclinical research that led to all these unsuccessful clinical trials has been carried out with models of brain cancer that are highly immunologically active. And so this doesn't represent the human disease at all. So a lot of the research that's happened has been on a really kind of fatally flawed system. And so I said 5 years ago, we should be using models that actually reflect human disease. And that's where I gave the problem to Devlin. So I introduced Devlin Forsythe, and he's going to tell you about what he's done to try and address this problem. Forward, backward.
Thank you. All right, probably the best introduction to my content I'm ever going to get. So yeah, I'm Devlin Forsythe. I've just finished my PhD at the end of last year, so I'm going to be giving an abbreviation, some highlights of my work. So we're taking a step back to what Melanie was talking about at the very end. My work is in the preclinical setting, so the lab work that goes into informing what we do in clinical trials, hopefully down the line. But taking a step back, we're going to talk about preclinical models. So in all cancers, but especially in glioblastoma, the models we use are critical. They help us to ask these important biological questions. Probably the most important one at the moment is, why are the clinical trials not working? So we can use these models to ask that question, try some new therapies, or we can ask something a bit more specific, like if the immune system is suppressed, that's stopping it, enhancing the tumour, why is it doing that? We can try and figure out these questions, the answers to the questions that we can't get from the human data, or we can try something completely new with these models. So we can make a new treatment that hasn't been tried in a different cancer, and try something new for glioblastoma. Importantly for this topic, animal models are essential for this kind of work. In order to develop and make a tumour that is as complex as we see in people, animals are the only way to do this kind of research. It's the only way to get all the cancer cells, the immune cells, the stromal cells, all in the same place. But in saying that, the welfare and the health of the mice and animals that we use is at the forefront of our mind. So we're making sure that, especially in cancer research, but in all animal work, the pain of the mice is limited as much as possible. And with this concept of the 3 Rs, where we're essentially, if it's possible to replace animal work with non-animal work, we will do that. But in situations where we need to, animal models are essentially the standard for preclinical research. But overall, the project that I got given was to develop this new model for GBM research, because clearly something new is needed to help research go forward. At the moment, the models which are really frequently used are not improving any of the outcomes. And I could probably give an hour presentation on why, but in the context of immune therapy, what we know is when we try all of the immune therapies that have gone to clinical trial and have been used in other cancers successfully, those are working in our mouse models. The mice are actually recovering successfully, and we know that's not the case when we try these drugs in people. So there's clearly a disconnect between the model and the human cancer. So to introduce the models that we are using, there's two major ones. The first, and probably the most frequently used, is the transplant model, where we take the tumorigenic tissue and we engraft it into the mouse, and then that will grow into a tumor that we can use to study. They're really fast, they're relatively cheap, and they form consistent tumors, which is what you want in a lab. But we know that these tumors don't contain the mutational profile, the genetics that we see in human tumors. And down the line, we know that this means that they do not contain the immune composition that we expect out of a human tumor. So the alternative to this is genetically engineered models, where you introduce the mutations to the mouse, and the mouse will then grow the tumor spontaneously. These are, as you're introducing the mutations, you know they're going to look like human tumors, because you're doing that work yourself. They have a bit more of the immune profile that you'd expect from people, but they're expensive and they're quite slow to grow. So not every lab will have the capability to introduce genetic modification in their facilities. So overall, we've got this kind of two completely polar opposite types of treatment, one that's not great but is easy and commonly used, one that's expensive. From this, we had this idea, can we kind of combine those two approaches together, do something new and make a new model? So that's what we did. We made this thing called the GemClem, or the genetically engineered mouse cell line model. So we took a mouse cell line, not a whole mouse, introduced mutations to it that we knew were in human glioblastoma, and then the idea was to take that cell line and use it for transplant. So kind of the best of both worlds. We're introducing known mutations and getting the speed and reliability of a transplant model. Overall, the main idea was to use this to study really specific issues and challenges. We could put a mutation in that we don't know what it's going to do and try and figure it out. But we could also, hopefully, our hypothesis was that through doing this a different way, we'd get a more accurate immune response into that tumor, and we could test out immune therapies in that kind of that relevant immune background that we're missing currently in the models. I made quite a range of these for my PhD, but I'm going to talk about one today, and that's one that models glioblastoma most accurately. We introduced mutations that are commonly seen, and so I'm going to be talking about one that P10 knockdown in RAS-V12. Just common mutations, but when I say that, that's the model I'm talking about. So the first kind of year or so of my work was kind of doing the more feasibility, will this model work? So here's an overview. I'm not going to be talking about the more cancer cell biology stuff, but this model reliably formed tumors. 100% of the time, tumors were developed. It formed fast tumors in line with the current gold standard in 3 weeks. The tumors we knew had the right mutational background, and importantly, these tumors were really aggressive, so in line with what you'd expect from this terrible human disease. So going forward from that information, we kind of had the idea, well, if it's working in the tumor cells, let's see what's going on beyond the tumor cells. So importantly for this talk, we wanted to look at the immune cells because we know that they're so important for helping that tumor progress and importantly for helping resist the immune therapy. So to do this analysis of the immune cells, I had two main tools. One was confocal microscopy and one was flow cytometry. They're kind of complementary techniques in the lab. Confocal microscopy, I can visually look at what immune cells are within the tumor, whereas flow cytometry is a lot more high throughput quantitative, so you can get a bit more data out of what you're looking at. So first, to introduce this very precise tumor, just description. In a tumour, we've got lots of different cells. We know we can guess what's in there, but in order to, instead of using light like a normal microscope, confocal microscopy, we can target cells that we want to look for with antibodies. And on top of those antibodies, we've got this fluorescent marker. And instead of using light, we're going to use a laser to target those antibodies, and that way we get fluorescence and pretty colours when the cell that we're looking for is in that tumour. And we can do this with multiple different antibodies, different colours, put a picture together where we're only going to see the things that we're staining for with our antibodies. So an example of this here, I've got on the left the gold standard, the currently used transplant model, and on the right is my P10-RAS. In red, I've stained for a marker called CD45, which is all immune cells. So what you're looking at here is just a confocal microscope image. Everything in red is an immune cell in the tumour. And just looking at this marker alone, we can see that both tumours do contain a lot of immune cells, which is good. We know this about tumours. And you could compare between the two of them and say that our model has a higher density. There is more immune cells within it. But alone, it's not going to do an awful lot. So this is why we introduce more markers. So in blue now, this is the nuclei of all the cells within the tumour on this section. So we get an idea of how crowded the tumour is, how many cells there are. And then we've got the nice little quirk of our model that the tumour cells are green. So on the right, we can see the green tumour cells. So this kind of analysis is really good for preliminary information. We know that there are immune cells, but also using confocal microscopy, we can ask more specific questions. So here we're looking at two different suppressive immune cells, two cells that are enhancing the tumour. So we've got in pink macrophages and in yellow microglia. They're quite similar cells, but doing a little bit different suppressive functions in the tumour. And you can see on the left that the gold standard model got a bit of both, balance of macrophages and microglia, whereas our model on the right, very heavy towards the macrophages. So these kind of visualization panels are really good, and we can make inferences based on this. It's a little bit limited because of the way you do microscopy is you take a very thin section and you're only looking at what the microscope can see. So in order to get an idea of the whole tumour, you'd have to either do a lot of sections with a lot of stains, or we can move on to our or we can look at them all together. We can look at them with a quantitative analysis of flow cytometry. So instead of looking them at the cells in a plane, we are going to break them up into single cells, into a cell suspension, do a similar staining protocol, and then we run them through a flow cytometer. So it's essentially just a really thin, narrow hose. When the cells come out the end, they come out one cell at a time in a droplet, and then we fire the laser. And that way we can get essentially the whole tumour analysed for every cell that's in it. Like microscopy, you have to know what you're looking for, so you have to put the antibodies on it. But we get high throughput, we get a lot of data out of this, and we can also look at multiple different antibodies on each cell. So you can know what type of cell it is, you can know whether it's going to help the tumour, whether it's going to suppress the immune system. You get a lot more out of this approach. You don't get pretty pictures with this one, we get a bit of data. So what we're going to look at here on the right is three of the main cell populations that we see in glioblastoma. Monocytic cells are suppressive, and then we've got our dendritic cells and our T cells. Those are the cells that you're wanting to get them revved up to target the tumour themselves. And what we can see is that in the left, in grey, is the standard model called GL261. There is a significantly higher number of dendritic cells and T cells than in our model. So we can see that there are less cells within our tumour than the current model that are able to target the tumour and kill it. On the other side, we've got our monocytic cells. There are more suppressive cells that are going to enhance the tumour growth in ours than in the current model. So on this comparison alone, we can see more suppression, which is what you see in human tumours, and less of that pro-tumour targeting immune response, and which is what we expect to see from our human if we were to make an accurate model. In terms of immune therapy, this means that if we were to use this model to trial out immune therapy, we would expect that we've got this low level of T cell, low level of dendritic cells. We need to get them fired up, ready to target the tumour. because there's less of them, we would expect it to be harder. We would expect immune therapies to struggle to work to effectively kill this tumour, and because of that high suppressive monocytic cells, we would expect that to make it harder as well. So, in conclusion, immune therapy has a lot of promise, and Melanie spoke about that with all the other cancers. It's definitely something that we should keep pursuing, but we know that it's tricky with glioblastoma. So really what we need to keep researching is why is it not working, and try a different way around it. My research, and this is really just a quick highlights of the key points, showed that we can model more accurately than the current models glioblastoma. We've got the mutation profile, we've got the right cells in there, and we know that there are suppressive cells, those macrophages, but we should really keep working to figure out what they're doing and use this model to do that. Future work should and will look into using immune therapies on this model, so use those T cells, CAR T cells or immune checkpoint inhibitors. And immediately, next steps in our group is that we're using this model to get some collaborations going with groups outside of Wellington, so Auckland, Australia, the US, everyone that we meet at conferences, we're telling them about this model to try and get different ideas on how to treat glioblastoma and use this model to try and answer questions. So this research hasn't just come out of nowhere. So in the years before my project, a lot of people put research in that helped get it off the ground. And a lot of people in the future are going to keep working on it. And additionally, a big thanks to Neurological Foundation for helping with the funding to get this project going. Thank you very much.